Hello and welcome to this webinar titled From Concept to Market, Unique Approaches in Biomanufacturing, hosted by Biopharma Asia magazine and presented by Dr. Jinkai Lee, Vice President of Woozy Biologics. This presentation will be followed by the next generation of single-use bioreactors up to 5,000 L of unprecedented performance, turndown and flexibility, presented by Kevin Mullen, Senior Product Manager of Single-Use Systems at Thermo Fisher Scientific. My name is Stephen Edwards and I will be your moderator. Before we begin, I would just like to inform our viewers that there will be a live question and answer session after both presentations near the end of the webinar. Audience members can send their questions in at any time during the webinar via the questions tab located directly below your webinar screen, and we'll go through these at the end of the webinar. Now please allow me to introduce our first presenter. Dr. Jinkai Jimmy Lee is currently Vice President of Woozy Biologics. Dr. Lee has over 17 years of experience in biologics process development, scale-up, and cGMP manufacturing. His group contributed to over 40 IND submissions, both in and outside of China, as well as BLA submission works. Dr. Lee joined Woozy Biologics in 2011, helped build and led the cell culture process development and pilot plant production group, and then managed the cGMP drug substance manufacturing facility. Before joining Woozy Biologics, Dr. Lee was a cell culture process development group leader at Eu Gnp Inc. Before that, Dr. Lee worked at Tanox Inc., where he led the manufacturing sciences group supporting full production of a MAB product, and at Diversa Corporation, now BASF, in charge of process development for multiple recombinant enzyme projects. Dr. Lee holds a BF degree from the Singhua University and PhD degree from the University of Maryland, Baltimore County, majoring in chemical and biochemical engineering. I will now be handing over to our first presenter. Welcome, Dr. Lee.
Thank you, Stephen. Hi, everyone. Welcome to the webinar. So in my presentation, I would like to introduce you WuXi Biologics approach in manufacturing, in biomanufacturing. Our goal is to be able to serve our clients from the concept stage to IND submission and all the way to commercial manufacturing. So I will go over the slide deck briefly, and Stephen can help me to advance the slides. So first, just a quick disclaimer. And then I would like to go over the outline of the talk briefly. I would begin by a quick introduction in terms of the biologics manufacturing challenges, and also go over the evolution of facility design. And then majority of the talk will be to introduce how WuXi Biologics approach biomanufacturing in trying to meet the needs from concept all the way to commercialization. And I will then end with a brief summary. So the next slide, this shows that in the new era of biologics, therapeutics, we can see that we now have many different modalities, from typical monoclonal antibodies to bispecific, fusion protein, antibody drug conjugate, or some recombinant proteins. The disease areas also vary quite a bit, especially with the immuno-oncology booming in the last couple of years. But regardless, the core biomanufacturing challenges remain the same, and that is to try to balance between cost, speed, quality, and the ever-increasing need for flexibility. The next slide. In terms of the challenges of biomanufacturing, we know that for biomanufacturing, technical challenge is always the important one, especially on product qualities. There's old saying that, process is the product, meaning that for biologics, if the process changes, even sometimes for tech transfers, your product quality could end up being different. So it is always a challenge to try to maintain a consistent product quality. The other interesting question that relates to the difficulties of biomanufacturing is illustrated by introduction of biosimilars. In recent years, there's more and more discussion about whether biosimilar can become interchangeable to the novel drugs. This was never an issue for small molecule drugs, but for biologics, it used to be that we can only call them biosimilars. So I think the fact that, as shown by the publication here, the fact that we are already talking about interchangeability on biosimilar does show that as an industry, we have advanced dramatically on understanding of product quality and how bioprocesses might impact that. Next slide shows the regulatory and compliance challenges on biologics manufacturing. We know that biologics manufacturing is heavily regulated. It is probably one of the most heavily regulated industries. And it doesn't help that very different regulatory agencies have their own, sometimes rather unique approaches or requirements on biomanufacturing. And this is illustrated by the recent introduction of the EU GMP Guide Nx1. It has been as a draft, to circulate around for a few years, but because of the extensive requirements on the facility and on GMP operation, there were many, many concerns and questions from the industry that, today it still has not been implemented. But it does, illustrate that, a regulatory body can have significant impact on how we run our biologics manufacturing. The next slide shows that the other ever-increasing pressure on biologics manufacturing is cost. So just last month, we saw a record-breaking drug price on CAR-T and gene therapy, right, costing $2.1 million for one single dose. And also, a few years ago, when PCSK9 antibody drug came to the market, the immediate question was how much would it sell? Because as a drug for treating chronic disease, obviously the cost and the price burden is an important topic. Over the last couple of years, with the emergence of China's biologics industry, the impact on cost and the discussion on cost has been increasing dramatically. As an example, we saw that China's own domestically manufactured PD-1 entered into the market at a dramatically lower price compared to what this sells in the U.S. Right? Basically about as low as 10% of the U.S. cost. And this definitely has led to a lot of discussions and debates, and the FDA commissioner has openly remarked that U.S. will welcome PD-1 from China in help to potentially bring down the drug price in the U.S. So I think, that cost pressure will only get even more and more significant for the future. So in terms of the biologic manufacturing facility, as recently as about 5 to 10 years ago, it used to be that most of the facilities are built for manufacturing of blockbuster drugs. So they typically will be single-purpose facility built with stainless steel bioreactors. And in those situations, cost was not a big concern because cost of goods will only account for about 5 to 10% of the drug's sale price. But this has dramatically changed over the past 5 to 10 years because the need for speed and flexibility, as well as the cost control from the government, has increased dramatically. So we also see more and more small market drugs, introduced by some of the CAR-T, personalized medicines, or orphan drugs. And with more and more small and mid-sized companies developing drugs focusing on certain small disease areas. The other need is the global supply chain to be able to manage the demand from various markets. At the same time, the facility operation approach is also evolving, mostly driven by the industry. Now, we see more and more facilities with multiple drugs being manufactured. And there's also discussion about concurrent manufacturing, meaning to manufacture 2 different drugs in the same suites at the same time. Next slide is a quick snapshot of what was published back in 2010 when some multinational companies, basically industry and users, proposed for multi-purpose use of manufacturing facilities. And that basically led to operation approach changes in many different drug companies, pharmaceutical companies. Next slide. So nowadays, we see that manufacturing facilities have more and more varieties. It used to be that most facilities are single-purpose, stainless steel-based. Now we see hybrid facilities and also disposable, completely disposable technology-based facilities. Next slide. So the way WuXi Biologics look at the ideal features of future facilities for biologics manufacturing contains the following characteristics. The first one is that it has to be a flexible facility, meaning that it can handle different cell culture modes, for example, fed batch, perfusion, or concentrated fed batch. It also needs to be able to handle different processes with different productivities, for example, from 1 gram per liter to maybe 10 gram per liter. Also, the facility has to be state-of-the-art to leverage the best available technologies, also with a sophisticated automation and data archiving capabilities, also electronic batch records. The facility should be also modular, and that when you need more capacity, you can simply scale out instead of scale up. And also you can build the same facilities in different locations. Also, the facility of the future has to be cost competitive so that we can control the cost of goods effectively. Right? So it comes down to a question of, for example, can you build a facility that can manufacture 1 ton of products, and the facility will only cost about $100 million? And the short answer to that is yes. WuXi Biologics did build that facility a couple of years ago. So next slide shows that the way we approach our manufacturing network is that we wanted to see the following 5 different characteristics. We wanted to implement single-use bioreactors, and we wanted to rely on scale out instead of scale up to meet different demands. And we wanted to also heavily invest in continuous manufacturing to increase the productivity and lower the cost. And we want our facility to be modular. And the other important feature is that we want to build a network of manufacturing facilities so that we can offer any client dual sourcing, meaning manufacturing of the same products in different facilities around the globe. So I will go over those one by one, some more briefly than the others. So the first is the use of single-use bioreactors. We probably don't need introduction on this, as by now most people are familiar with the technology. We go to the next slide. Basically, in WuXi Biologics, we have only utilized single-use bioreactors. Up to now, we have not used any stainless steel bioreactors. And we were able to do that by manufacturing almost 500 batches with a success rate of 98%. And relying on single-use bioreactors also allow us to dramatically lower down the CapEx investments and be able to build our facilities much faster than with stainless steel technologies. Next slide. This is an example of a 4,000 liter bioreactor scalability data. We are one of the earliest adopters of the 4,000 liter bioreactor, and this basically shows that we have excellent scalability between 250 liter to 4,000 liter. Next slide. There are obviously still challenges on using disposable bioreactors. It used to be that scale limitation is what people talk about the most. But with the introduction of 4,000 liter and now also 6,000 liter from ABAC and then 5,000 liter from Thermo, I think the question is, will that ever become a limit? Can we potentially see single-use bioreactors even bigger scale? And if that becomes reality, capacity or limitation in size will not become an issue at all. Obviously, a manual installation of the single-use bioreactors, especially on some of the traditional pros, is still somewhat challenging. And from time to time, we will see cell lines that are being sensitive to bags. So those need to be taken into consideration when selecting the cell lines and doing process development. And one of the most important aspects of relying on single-use bioreactor is the dependence on our suppliers. And we feel that as much as the situation has improved, we still hope that our suppliers can continue to improve to ensure a robust supply of the single-use products. In terms of our approach to meeting the demand for blockbuster, megaton products, our approach is to using scale-out strategy and continuous manufacturing. And I will introduce those in the following slides. Next slide. This is a simple concept of scaling out versus the traditional scaling up. It used to be that with stainless steel bioreactors, you only can go up in the scale when you get into different phases of the program. And that has always been a challenge for cell culture. But with stainless steel bioreactor, with single-use bioreactors, we can simply increase the number of units when the demand increases. So for example, what this illustrates is that we can start with one single 2,000 liter bioreactor for IND and for early clinical, and then we can increase the number of bioreactors to 3 or 6 or even 12 as the program and when it gets into commercial manufacturing as the demand increases. Or in some case, when the demand drops down, we can reduce the number of bioreactors as well. So that's the essence of the scaling out approach. Next slide shows that we do actually see many advantages of doing the scale out. Although the size limitation is a real reality that we have to rely on scale out, but it also does offer many advantages. For example, you can utilize the same 2,000 liter bioreactor all the way from pre-IND manufacturing to commercial manufacturing. And by doing so, you eliminate the risk of scaling up that's always associated with the standard steel bioreactors. It also allows you a lot of flexibility in terms of thinking about how many bioreactors you might need at different stages of the product life cycle. We don't need to think about building different facilities because you can only decide, you just need to decide how many bioreactors you would like to operate at different stages of the program. Next slide. So this is the table basically illustrates that if you have a product that needs 100 kilograms per year, and depends on your productivity, you will need different number of batches per year. For stainless steel, if the scale is 10,000 liter, you will need 14 batches if the titer is 1 gram per liter. If we use only one 2,000 liter bioreactor, that means we have to manufacture 70 batches a year, and that's a lot of batches to run. But with the scale out approach, if we, for example, use 5 times 2,000 liter, which is equivalent of one single 10,000 liter stainless steel, then your number of batches basically is the same as one single stainless steel bioreactor. Now, what's shown in the bottom row, color in red, is that if we have 4,000 liter single-use bioreactor, the scale out becomes even easier. You would just need 2 by 4,000 liter bioreactors to be almost equivalent to the 10,000 liter stainless steel. Next slide shows a similar concept in that with different disease areas, with different demands of the drug, if you rely on stainless steel facility, then you need to move to different facilities, to different scales for different demand. But with single-use bioreactor, with the scale-out approach, you can manage it by just using different number of the bioreactors. Next slide shows basically how we can operate this scale-out approach. This is an example of what we call a 6-pack. This is in our manufacturing 2 facility where we have 14 by 2,000 liter bioreactors. We can run the operation with a scale of 6 by 2,000, equivalent of 12,000 a single batch, and you can run alternate suites in alternate weeks. And you just need one single downstream train to support the 2 upstream trains. The next slide shows that if you rather stay with 3 by 2,000 liter, you can do that. You can run it with 2 protein A, but then you can pour the products after protein A step, and it will continue downstream, and it will still be one single batch for basically 6 by 2,000 liter. So separated by protein A, but then pooling after protein A to have one single batch equivalent of 12,000 liter. Next slide. So switch the topic a little bit to continuous bioprocessing. I think The drivers are also clear in that, especially over the past 5 years or so, there's been a lot of discussion on the benefits of continuous bioprocessing, with the key benefit that you can use a modest scale of the facility to support a variety of different demands of different products. Next slide shows the schematic of the continuous upstream processing, nowadays mostly supported by the use of the ATF device. And the next slide, at WuXi Biologics, we also curtail the perfusion process slightly. We call it WuXi Up. Basically, it's a custom designed perfusion process with a duration that's similar to fed batch. So we basically run the perfusion process for 2 to 3 weeks, but at ultra high productivity. And what's highlighted in the box is that for a fusion protein, we can generate up to equivalent of 51 grams per liter bioreactor volume with just a 20-day continuous process. So that's easily 5 to 10 times more productive than a traditional fed batch operation. Next slide shows a case study that illustrates that this is for the same cell line with the same cell culture media and controls. When you run it at fed batch, a title of 2.8 grams per liter, by using the WuXi Up process, we can obtain over 50 grams per liter productivity with a similar culture duration, a slightly longer culture duration. The next slide shows a calculation, a simple illustration of the calculation that you can use a 1,000 liter bioreactor to achieve equivalent of 10,000 liter fed batch scale. And that can basically supply you over 1 metric ton of the products each year. Next slide. So I would like to turn to the concept of meeting the demands by providing global dual supply. So we have, what's shown in this slide is that we have a very unique pipeline in WuXi Biologics service. Up to June of 2019, we have a total of over 224 molecules in WuXi Biologics service, and among them, 58 of them are actually first in class. So they have a combination of programs in different stages, and with the expectation that many of those will move to late stage and being able to enter into commercial markets, we wanted to see how we can best support those with our manufacturing network. So next slide shows the technologies that enable us to develop our global supply chain. And the enabling technologies are the WuXi Body bispecific platform, the WuXi cell line approach that can support up to 60 different cell lines a year, and then the ultra high productivity WuXi Up process that can generate up to 50 grams per liter equivalent. Next slide shows that over the past couple of years, we have put together a plan to develop a robust global supply chain with the projection that by 2022, we will have a total capacity of 280,000 liters of cell culture capacity. This is going to be distributed mostly in US, Europe, and China, as well as in Singapore. The next slide shows the detailed list of our drug substance manufacturing facilities. Highlighted in the dashed docs are the ones that will come to the market starting in 2020. So you can see that our facilities cover a few different kind of operations: fed batch, perfusion, or what we call the concentrated fed batch, or IPC. Also highlighted in dark red are some of the unique approaches we put together for our facilities. For example, on MFG 7, MFG 8, and MFG 12, our plan is that we can install either 12 by 4,000 liter bioreactors, or we can actually switch those to 2,000 liter bioreactors. This is what we call the plug and play. Using the same set of controllers, we can control either the 4,000 liter bioreactors or 2,000 liter bioreactors, thus offering additional flexibility to our clients. Next slide basically highlights the various different cell culture manufacturing operation modes, and then the corresponding facilities to support those. So as you can see, from 2,000 liter fed batch to 4,000 liter fed batch to perfusion and to CFB or IPC, for each cell culture mode, we offer a mid-scale facility or large-scale facility across China, US, and Europe, or Singapore. So this provides a robust global supply chain with at least dual supply capability for every single cell culture mode. The next slide shows a quick example of how this might translate to a real situation. For example, we can start preclinical and early clinical manufacturing in our Nx1 facility with one single 2,000 liter bioreactors. And then as the program moves forward to PPQ, we can move to manufacturing 2 and use our scale out approach, now doing 3 by 2,000 liter for PPQ, and launch the product into the market. Once the products are in the market, we can further expand from MFG2 to MFG7 in Ireland, and we can increase the bioreactor from 3 to 6 or even 12, thus truly offering the global supply for important molecules like this one. The next page is an example quite similar, but with 4,000 liter bioreactors. We can start with our MFG4 facility for preclinical and then early clinical, and then move to MFG5 for PBQ and commercial launch with, for example, 3 by 4,000 liter bioreactors. Once the products are in the market, we can further expand to Ireland again, thus having global dual supply in MFG7 and MFG5. So with that, I'd like to move to the final slide and basically quickly conclude the presentation. I hope I can already demonstrate that in today's manufacturing, flexibility is the key word. We need our facility to be flexible to meet the demands of ever-changing needs. Also, WuXi Biologics has demonstrated that we can rely 100% on disposable bioreactors, which can now go up to 4,000 or even bigger scale. And it can become a viable solution for commercial manufacturing. Also, I hope I can already show that our approach of scaling out instead of the traditional scale up is a viable solution to provide maximum flexibility and also consistent product quality for biomanufacturing. And last but not least, continuous processing offers a great promising future for biomanufacturing. So with that, I'd like to conclude the talk and thank you for your attention. Steve?
Thank you,
Stephen?
Dr. Lee. Now, before I introduce our next presenter, Kevin Mullen, once again, I would like to remind our viewers that there will be a live question and answer session at the end of the webinar. So please feel free to send in your questions at any time during the webinar via the questions tab located directly below your webinar screen. And we'll go through these at the end of the presentation. Now, please allow me to introduce our second speaker. Kevin Mullen is a Senior Product Manager at Thermo Fisher Scientific, based in Logan, Utah. For the past 2.5 years, Kevin has been managing the single-use bioreactors and single-use fermenters portfolio. In his role, Kevin is working closely with the development team to enhance and better characterize the performance attributes of single-use hardware systems. Prior to joining Thermo Fisher, he spent 13 years as an engineer and product manager in the medical device industry. Kevin holds an MS degree and an MBA from Utah State University, majoring in biological engineering at business respectively. I will now be handing over to our second presenter. Welcome, Kevin.
Thank you, Stephen, and thank you, Dr. Lee. A great presentation. I appreciate you sharing all of your insights there at WuXi. Hello, everyone. My name is Kevin Mullen. Today, I will be presenting on our next generation of single-use bioreactors that we are working on at Thermo Fisher Scientific. I need to acknowledge a colleague of mine, Dr. Mark Smith, who presented on our next generation reactors for the first time at the BPI East last week in Boston. And I'm excited to be able to share this content with you on this webinar. Just as a disclaimer, I will be showing some data about our next generation reactor. Some of this data is preliminary and may be updated in the future as we continue to do some characterization studies. So first of all, I want to talk about the needs for a next generation single-use bioreactor. Dr. Lee did a great job talking about what they're doing at WuXi as far as intensifying their cell culture and building out and scaling out. And he also did a great job introducing us to how they're using complete single-use manufacturing sites. And throughout the last 15 years, single-use bioreactors have been adopted within our industry. There's been a lot of progress made on how the bioreactors are used. And there has been some limitations that have been known among single-use bioreactors that we have learned about, and that has facilitated us to look at a next-generation single-use bioreactor to overcome some of those limitations. One of the first ones is intensified cell culture. Current bioreactors can do intensified cell culture, but it is a challenge sometimes maintaining the power and mass transfer needed to support the high cell densities that we are now seeing in intensified cell culture. Dr. Lee presented that they're up to close to 100 million cells per milliliter, and we have some customers of ours that have expressed that they would like to go above 100 million cells per milliliter, and that does require more power and more KLA capability. The next trend that we're seeing in industry, and this is true for single use, and Dr. Lee presented this very well, is the need for flexible and scalable facilities. That's one thing where single use really thrives, to be able to have flexible and multi-product facilities, and we want to maintain that capability. The third thing is capacity demand. We're seeing a large push for more capacity within our industry, especially within single use. There's been quite a few clinical molecules that have been developed within single use, and now they're going to commercial phase. And when you're at commercial phase, some of those molecules are transitioning out of single use and into stainless steel just because there hasn't been capacity within single use to be able to maintain that. So there has been a need for having a larger than 2,000 liter bioreactors available within our market. So with that being said, I want to introduce you to our next generation of single-use bioreactors, which we're calling the High-Performa Dynadrive. So the High-Performa has a great name amongst our industry. We use the High-Performa name for our existing bioreactors that range from 50 liters up to 2,000 liters. But now this new Dynadrive will take us from 50 liters up to 5,000 liters. So not only did we want to increase the volume capacity within single-use bioreactors, we wanted to increase the performance. So we wanted to have the best-in-class power, mixing, and mass transfer. And I'll cover some of that in more detail on subsequent slides. We wanted to maintain an efficient facility footprint at those scales. So we wanted these single-use bioreactors to take up as minimal footprint space as possible, and so that they can be easily implemented into existing facilities and not take up very much space, and they can be implemented quickly. Also with these reactors, they'll have the best-in-class seed train. That will enable what we call N-K seed train capability. And they all will have at least 10-to-1 in-vessel turndown. So the 50 liter would be able to run at a 10-to-1 volume, meaning you could seed it at 5 liters. And even the larger sizes, like the 500 and the 5,000, will allow us, we can have a 20-to-1 turndown ratio in those, meaning that you could operate a 500 liter reactor with as little as 25 liters. We wanted to have the ease of use of ergonomics. We recognize that the 5,000 liter bioreactors could be intimidating to load a BPC, and so we took special design considerations to be able to automate that BPC loading as much as possible. And we want to be able to load it with 2 operators in less than 30 minutes. And we want to maintain flexibility for custom application and future needs. So we've designed the hardware to have as much open space as possible, whether it be open space that is closed with, say, plexiglass or plastic, where we can modify that plastic easier than we can modify the stainless steel, so that we can adapt for future applications that may come up with, as far as having ports needed for perfusion or various probes throughout the reactor. We can adapt to that quite quickly. And you'll see there at the bottom the dates for when these new reactors will become available. So our smaller sizes we will be launching in 2020. The 50 liter will be quite soon in 2020, 500 liter around mid-year. And then our larger sizes, our 3,000 liter and 5,000 liter sizes, will be in either late 2020 or early 2021 as far as their commercial availability. So what is the Dynadrive technology? What makes it different? Most of you are probably familiar with the current Thermo Fisher high-performance single-use bioreactors that have a single impeller. When we were thinking about upgrading and taking the volume to the 5,000 liter scale, we realized that we needed to design the impellers differently. And pictured here is an example of what the impellers look like and how they are supported inside of the BPC. So as you can see, we have multiple impellers. These multiple impellers allow for us to have very good power. So our power per volume can exceed greater than 80 watts per meter cube, while also maintaining relatively low tip speeds, which will help maintain a low shear environment for the cells. Also, that drivetrain has a very low impeller at the very bottom of the BPC, which enables our incredible turndown ratio of at least 10:1. These multiple impellers, along with the shape of the reactor, allow for great mixing times. We see mixing times of less than 45 seconds. And this helps our KLA performance to have greater than 40 inverse hours of mass transfer performance. Maintain the bubble residence time inside the reactor for quite a long time due to our mixing capability. And also we've designed the spargers to produce bubbles that are of the most ideal size for your oxygen mass transfer as well as your CO2 stripping. We will be maintaining the robust and reliable film that Thermo Fisher uses. We'll be using the Thermo Fisher Aegis film with this new reactor. And we will have extensive lifetime testing on this drivetrain performance. And we engineered the BPC and the hardware with ease of use in mind. And we've learned a lot over the last 15 years since we've been selling single-use bioreactors. And one of the things we wanted to improve upon was the ergonomics and reliability of loading those BPCs in the hardware. Along with this design, it allows for some flexibility for volume ranges. I mentioned the low seed train turndown ratio, but also due to the design of the impellers and the location of the impellers, the reactors will be able to be used at various terminal volumes, meaning you don't always have to run at full volume for your terminal volume. You could run it as low as 1,500 liters or 2,000 liters, and the same thing is true with the 5,000 liters. So it allows you some flexibility within the reactor to accommodate various batch sizes that you may need. And here's a little animation of what the drivetrain looks like. Rotates, all the impellers rotate at the same time, and it rotates together. When it actually rotates, it forms a bit of a helical shape, and that gives us great strength and robustness while also having the drivetrain be flexible for packaging. So now I'm going to talk a little bit about the impeller configuration. So particularly at the large size, like the 5,000 liter size, we recognize that there is a need for multiple impellers. The graph there shows impeller count matters. So the green line shows that we get very good mixing time at a relative low power. If we removed one of the 3 impellers that we plan on, then we needed to have 40 times the power go to get the same mixing time. If we removed 2 impellers, we needed 60 times the power to get the same mixing time. And so the multiple impellers definitely help reduce the mixing time and also provide great power into the system, which you can either utilize that high power for high cell density cultures to properly mix them, or you can reduce the RPM and help make sure that you reduce the tip speed and which prevents shear, cavitation, and breakage of your cells. Also, we took a careful note on where the impeller locations should be. We made sure to distribute them in a way that they would be distributed power within the reactor and provide as good a performance as we can within the reactor. And we do really get some great performance with these multiple impellers. We also looked at the geometry. As many of you know, there are many approaches to mixing and different geometrical approaches towards mixing. There's a centered cylindrical approach, which it does form a vortex, and you get unidirectional flow, which leads to pore mixing. Then there's the off-centered angled, which is what we currently use with our high-performer reactors, which performs very well. That does reduce the vortex. You do get unidirectional flow, but it does improve it compared to a centered cylindrical impeller. And then there's baffled, which some suppliers provide, and baffled within single use is quite difficult because you need your bag to conform around those baffles, or you need to include the baffles as part of the bag itself. Usually with baffles you don't get a vortex, and you get disrupted flow patterns and good mixing times. But as you probably imagine from those first pictures I showed you, this new Dynadrive reactor has a bit of a different shape. It's a cuboid off-centered shape, meaning that the shape itself is more like a square or a cube, and the impellers are off-centered. We found that this gives no vortex formation, which we definitely want within our bioreactors, and it gives a very disrupted flow pattern within the mixing, which we want. And so you would get very good superior mixing times due to the shape of this reactor. And the corners act as baffles, which help distribute the mixing within the reactor. Also, the geometry, not only does it improve the mixing, but it also improves the volume per floor space. So on this slide, we're showing you that we get 27% more volume per floor space by being cuboidal in shape. One reason why we have a 3,000 liter Dynadrive is because that will fit in spaces where our current 2,000 liter bioreactor fit. So it uses the same footprint and the same height. And we recognize that customers will see the advantages of this Dynadrive technology, but may not have a facility that can house a 5,000 liter right away. But they do have a facility that could house a 2,000 liter. Therefore, we have this 3,000 liter that could be used, and instantly you can get 50% more capacity by using this 3,000 liter. Also, the shape improves our mass transfer efficiency and our bubble residence time. So we did look carefully at the column height and made sure that we could have as high of a column height as possible to increase the bubble residence time, which improves our KLA performance. Also, the sparger to impeller interactions is key when we talk about mass transfer. And as I previously mentioned, we can have up to a 20-to-1 turndown ratio at the 500 liter, 3,000, and 5,000 liter scale. So now switching gears a little bit to the mass transfer. So mass transfer isn't only dependent upon the great mixing or the shape of the reactor, but it has to do also with the sparging. So Thermo Fisher, back in 2014, introduced the drill hold sparge. And drill hold sparge is actually maybe a poor name for it. It's actually a laser precision drill hold sparge, where that laser, each of these spargers are made with precision lasers that cut holes of the same size and in a pattern and quantity that we have specified. And so because the holes are all the same size, we get uniform bubble size. And the holes are actually cut to different sizes depending on the size of the reactor you are using. And so the bubble size scales with the height and volume of the reactor. The great thing about a drill hold sparge is you get linear performance as far as your KLA in relationship to your vessel volume per minute flow rate. And therefore you can minimize the gas entrance velocity if you and dial in the KLA performance that you need. If we minimize the gas entrance velocity and the gas going into the reactor, that can also reduce the foam formation inside of your reactor. So in the graph there on the right, the red line represents a microsparge or a frit plus a DHS. And you can see that red line, you get quite a bit of KLA from the frit itself to a certain point, and then it plateaus. And that is not linear, so it's hard to determine exactly what KLA you will be getting at various flow rates. Whereas the black line represents the DHS alone, and that you can see is really linear and allows for great characterization of your reactor using that linearity and dialing in the vessel volume per minute flow rate to achieve the KLA that you need. So we're utilizing that drill hold sparge in this Dynadrive reactor as much as possible. But we've enhanced the drill hold sparge from what it has been. We've actually reduced the hole size so we get some slightly smaller bubbles and increased the number of pores, and so we can get more pores, and that increases our KLA performance. So that's represented here on this slide. So we're getting more than double the KLA performance in the Dynadrive than we have with our traditional bioreactors, while maintaining good carbon dioxide removal. So in the graph there on the left, you can see our traditional reactors, or what we call legacy reactors now, are in the dashed lines, and the solid lines represent the Dynadrive. And we tried to compare these at the relative powers that you can see. The Dynadrive in blue had a power of 18 watts per meter cubed, and the dashed line was 19 watts per meter cubed. So very, very similar. So you can see that solid line is about double what that dashed line is, as well as a greater power, 48 compared to 42. We get even more KLA performance using the Dynadrive. And then moving to the graph on the right, this is a very important graph because this is comparing our 50-liter Dynadrive and our 5,000-liter Dynadrive, and also our current legacy 2,000-liter single-use bioreactor. You can see the Dynadrive systems have more KLA than the current 2,000-liter reactor, but more importantly, the 50-liter and the 5,000-liter KLA performance are not only linear, but they lie right on top of each other, meaning that the 50-liter represents the 5,000-liter very well, and we anticipate and know that the 50-liter will be a great scale-down model of the 5,000-liter, which is very important when we talk about the reactors. Not only do you want to scale up to say 5,000 liters, but you want your scale down models to be very representative, so you can do a lot of your PD work at as small a scale as possible and know that it's representative of your large scale reactors. Here's some more data that we've generated with the Dynadrive. So the graph on the right compares the 50 liter, the 500 liter, and the 5,000 liter. And this is all at 20 watts per meter cubed and showing the relative KLA compared to the flow rate in your sparger. And then also we want to have scalability within a reactor. And so the graph on the right shows your red and green lines. The red lines represent the performance at different volumes within the reactor relative to different powers. So you can see the dashed line. The red dashed line has 30.5 watts per meter cubed compared to 24 watts per meter cubed at full volume. Oh, sorry, the solid lines are full volume and the dashed lines are the 30 liters within the same reactor and at various volumes. But you can see that we get close to the same KLA whether or not we're at 30 liters or at 50 liters within the same reactor, so we can have some flexibility within the reactor itself. Now a little bit more about the shape or height of the reactor. So this slide compares our legacy systems, shown there in yellow, and showing that. So our existing reactors go from 50 liters up to 2,000 liters. The 2,000 liter today requires a ceiling height of 3.8 meters. This new Dynadrive system, partly because it's cuboidal in shape, and because the drive system is all maintained in the BPC, you no longer need a drive shaft. We can actually fit the 3,000 liter in the same ceiling height, and as I mentioned, in the same footprint. Therefore, you can utilize the 3,000 liter in a facility that currently is built for a 2,000 liter reactor. The 5,000 liter will be taller than the 3,000 liter, and it may be more of a greenfield opportunity for you as you're considering expanding out to a 5,000 liter bioreactor. An important note that we wanted to make here is packaging. So when you're dealing with a 5,000 liter reactor, that the BPC or bag can be quite large. And so we took special account of that and wanted to reduce packaging as much as possible. And our drivetrain, our impellers, allow us to do that. It's a flexible system, and we can fit the packaging on a standard US or European pallet that is no more than 1 meter tall. And so that is important to consider as far as warehouse space of where you're going to store the BPCs prior to use, and making sure that the small packaging and handling can be achievable within your facility. Now I want to talk a little bit about the benefits of a high turn down capability in the reactor. So the 2-to-1 turn down, you can see that flow showing that it takes about 5 steps within your biosafety cabinet, which is quite risky. And this flow that we're showing is you have your cell bank, then you're going into a flask, which is done in an incubator. So we're showing 2 steps of that, and then you may go to a rocker reactor, and then to a 50 liter, then to 250, and then to say a 2,000 liter. So that flow has 5 biosafety cabinet operations and 9 tubing connections, as well as 4 BPCs. Thermo Fisher currently sells on our legacy system a 5-to-1 turn-down ratio, which helps reduce the number of vessels, thus reducing the number of BPCs. But it also requires some more steps within a biosafety cabinet. The 10-to-1 turn-down ratio in the Dynadrive allows for fewer steps in a biosafety cabinet, fewer tubing connections, and the fewer BPCs, as well as increasing your volume by 1.5 times if you end in a 3-liter or 3,000-liter bioreactor, or it increases the volume by 2.5 times if you end up in a 5,000-liter reactor. So there's a lot of benefit to the great turn-down capability of going to this 10-to-1 turn-down within the new Dynadrive reactor. So in summary, on this new system, we're still working on it. We do have some beta testing going on, but it is coming soon. And so we wanted to announce this to the market, and so people could be thinking about this, planning for it, and getting ready to schedule some demos throughout 2020. But the great advantages of the Dynadrive system is that we can scale up to the 5,000-liter now, and we can provide the great power. So as Jimmy alluded to, Dr. Lee alluded to in his presentation, they see great advantages at larger scale. They're utilizing a 4,000-liter reactor, and we'll be looking for larger, so this 5,000 liters is larger than the 4,000-liter. It also, they're utilizing perfusion, and they need more power within the perfusion capability. So not only do we provide more power and mixing, we have a great mass transfer, and we have great scalability of that mass transfer within the family of reactors. So from the 50-liter up to the 5,000-liter, we've been able to demonstrate that scalability. We have 10-to-1 turn down, which will reduce your seed train as far as the number of transfers that you need to make, and therefore reducing risk. We have performance volume ranges that reduce the number of PD vessels that you need. You can do a lot of different volumes within the 50-liter or 500-liter vessels, and therefore you can reduce the number of PD vessels that you need. We've designed it to be ergonomically robust and friendly, so that the BPC loading is efficient and can be done routinely, and therefore you know that it can be done reliably. And we do have a website available, so if you want more information, you can go to thermofisher.com/Dynadrive, and there you can find more information and request more information. So that's the end of my presentation, and I'll turn the time back over to, before that, I want to acknowledge our project team and alpha and beta testers that have been testing the Dynadrive thus far. So with that, I want to turn the time back over to Stephen and open it up for questions.
Thank you for that presentation, Kevin. As you just mentioned, now we will begin the question and answer segment of the webinar. But once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now our first question is, do you see the need to build stainless steel bioreactor facilities at some point? And that question is for you, Dr. Lee.
Thank you, Stephen. Now, I think, with the ever-increasing scale of disposable bioreactors, as Kevin mentioned about 5,000 liter coming out, and even 6,000 liter, I think potentially in the future, we might see even larger scale. And also with the increasing productivity of the modern day processes, I would think that we can supply commercial demands by solely rely on single-use bioreactors. So at least, this is the way we look at it from WuXi Biologics. We do not have plans to build any stainless steel bioreactor facilities.
Thank you. Our next question is, Thermo Fisher has large presence with their current hyper-perform single-use bioreactors. Will Thermo Fisher discontinue their current lines of single-use bioreactors? And that question is obviously for you, Kevin.
Yeah, no, we don't have plans of discontinuing the high-performa and the single-use bioreactors. Those are great reactors. They perform well, and depending on the situation, they should still be used, and they will be made available as needed even after the Dynadrive is launched. So we'll have the 2 lines of single-use bioreactors, and we will maintain those for the foreseeable future.
Great. Moving on with the next question. With the various process options, fed batch, perfusion, IPC, etc., how should a product company decide what approach to use? Kevin, shall I answer this one? Sorry, Dr. Lee, shall I answer this one?
Oh, okay. Thank you, Stephen. Yeah, I think nowadays, as mentioned, we do have a variety of options for the process. Ideally, a product development company we wanted to be able to choose the manufacturing process early on, so as early as possible. But it is also feasible to change to a different cell culture mode in the clinical stages of the program, with the assumption that you can demonstrate product compatibility so that there will not be any impact on the efficacy and safety of the drug.
Thank you. Our next question is, again for you, Dr. Lee. Do you see the single-use bioreactors getting even bigger?
Right, yeah. So as I mentioned in the first question, I do believe that, I'm optimistic that we can get even bigger. Until a few years ago, everybody thought that 2,000 liter is the limit of the single-use bioreactor scale. But, over the span of just a few years, we now already see more than double, in Thermo Fisher's case, more than double to 5,000 liter. So I think with the advance of the technology, I do see that potentially we can get even larger scale in the future.
Great. Our next questions, based on Dynadrive, so for you, Kevin, the Dynadrive single-use bioreactor looks interesting. How can we learn more about the Dynadrive?
So at this time, because we are just announcing it, the best place to go would be on the website, thermofisher.com/Dynadrive. There actually is a form there for you to fill out with a few questions, and that will lead us to make sure that we can direct our commercial team to come and talk to you and present some more information about it. So that's the best place to go right now is to the website. And
on the back of that question, what cell lines have been tested in Dynadrive thus far?
Yeah, thus far we've focused on the CHO. We've tested quite a few different processes with CHO cell lines. We continue to do quite a bit more testing, but we have done everything with CHO thus far.
Thank you. Our next question. Can Woozy disposable bioreactors be utilized for microbial fermentations? Dr. Lee, if I can answer that.
Yeah, thank you, Stephen. The facilities that we show on the presentation are all for mammalian cell cultures. So they are not available for microbial fermentation. But the technology is definitely available. Multiple suppliers offer disposable bioreactors for microbial fermentation.
Great.
And
Thank you.
Also, I wanted to add that also WuXi Biologics also has the plan to open our microbial fermentation facility in the near future.
And with that, thank you for your answers, and thank you, audience members, for your questions. Before I finish the webinar, I'd like to ask our presenters, Dr. Lee and Kevin, if you have any closing remarks. So, Dr. Lee, do you have any closing remarks for our audience?
Thank you, Stephen. So hopefully the presentation is informative to our audience, and we definitely welcome any contact if there's additional questions.
And Kevin, do you have any closing remarks for our audience members?
Yeah, thank you, Stephen, and thank you, Dr. Lee. I think the presentations went very well. I would also welcome any more feedback. You can send email to me directly, kevin.mullen@thermofisher.com, or you can reach out to your local sales representatives within Thermo Fisher if you have other questions regarding any of our bioreactors. So Dr. Lee talked about using single-use bioreactors within their facility, and I introduced our next generation bioreactor. Feel free to reach out if you have questions, and I hope this was just a bit of an introduction for you and informative for you. Thank you for attending.
And I would like to take this opportunity to thank our presenters. Once again, we had Dr. Jinkai Lee, Vice President of Woozy Biologics, and Kevin Mullen, Senior Product Manager of Single-Use Systems at Thermo Fisher Scientific. Thank you for sharing your knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send in your questions over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers sent back to you. Thank you, everyone. Have a great day.